Hello everyone, welcome back for more biotechnology. This is going to be a very quick video, but I just wanted to say a couple of things about what you tend to do with transformed cells after the transformation is done. So, what you're going to do with your transformed cells now that you have some sort of affirmation that the transformation went well, you can see your protein product, whether it's GFP or beta-galactosidase or something else. If you're confident that your transformation went well, what do you do next, right? So in the PGLO lab that we did, basically we looked at the GFP. It was nice to look at. It's pretty. It's nice to look at, but doesn't really have a whole lot of practical utility on its own. So if you're actually doing something that's going to matter a little bit, such as expressing a recombinant protein like insulin or TPA, something that has some sort of therapeutic or other practical purpose, the next step is going to be to take your transformed cells and to somehow isolate and purify those proteins so that they can be sold on their own. But the main problem that we run into right away is that those cells that are growing on the agar in the petri dish, they're not actually making a whole lot of protein product. Or I guess the more accurate way of putting it is that there are so few cells there that we wouldn't be able to get a whole lot of protein out of it, at least not enough for us to work with. So therefore, usually what's going to happen next is a scaling up process, meaning that we need to take those transformed cells and we need to grow a whole lot of them. We need to be growing so many of these transformed cells that when we harvest them, we are therefore going to get a very large amount of protein from a very large quantity of cells. So what this scale-up process is going to entail is basically taking a step-by-step approach to deal with the unique way in which cells tend to grow and the conditions in which cells tend to grow, which we'll have something to say about here in just a minute. But what we're going to do is we're going to grow our transformed bacteria in typically very large bioreactors. You can see some smaller ones here on the left that are actually contained within a 37-degree incubator. So these look like, they're maybe somewhere between 1 and 2 liters. So that might be kind of an initial starting point, growing up about 1 liter of bacterial culture. But eventually you may end up with a very, very, very large bioreactor that contains thousands upon thousands of liters of bacterial culture. So you can actually see what one of those looks like here on the right. So basically, the general idea is the more cells that we are growing in suspension, the more protein that we can get. So these are the sorts of things that biotech companies spend many, many, many R&D hours and dollars on researching to find the absolute optimum conditions upon which a lot of pure protein product can actually be manufactured and then marketed and sold. So what we're going to find out in the next video is that cells tend to stop growing and stop making proteins, therefore, when the cell density gets too high. So if we're growing bacteria in, say, a 1 liter flask, well, we're going to be growing and growing and growing bacteria, but eventually there are going to be so many bacteria in the flask that we don't really have room to grow anymore for several reasons. So basically, once we get to a point where a bioreactor has a maximum number of allowable bacteria, we're going to move on to the next step in the scale-up process. So if we're at a 1 liter flask, then we may next move on to a 5 liter flask. And then after we get maximum cell growth there, we may move on to a 20 liter flask, and so on and so forth. So we just keep scaling this process up, in which we take a culture that has a maximum number of bacterial cells, and then we transfer it into a larger volume of broth, and we give them a new opportunity to continue growing and to therefore continue making our protein that we want. Another thing that is worth mentioning, and as you can imagine, this can be very challenging, we need to maintain absolutely pristine sterile conditions in these bioreactors at every step of the way. If one contaminating cell ends up in your bioreactor, then we are essentially providing conditions for that cell to grow, and it can overtake our transformed cells and it can ruin the entire process. So we absolutely need to make sure that everything is completely sterile. And then another thing that's worth mentioning is that these sorts of things don't just happen on their own, so we are going to have technicians that are constantly checking these bioreactors to make sure that nutrient conditions are right, cell growth conditions are right, the temperature is what it's supposed to be. And most importantly, we want to make sure that these cells are indeed making our protein of choice. So you can imagine that an ELISA assay would be a very quick and easy way to just verify that your cells are making what they are supposed to be making, provided that you have a primary antibody against your protein of interest. All right, so that's going to do it for that very short video on the scale-up process after transformation. So join us next time and we will start talking about a couple more specifics about growing cells, cell growth kinetics, and other practical purposes, basically what you do after the transformation process. So thanks again for your attention, and I will see you next time. Bye-bye.